Hello everyone, welcome to Medverse, and today we are starting off with Thanatology, that is study of death. So study of death in all respects is known as Thanatology. Okay, Thanatology. The study of death in all respects. Study of death can be due to cessation of circulation, respiration, and brain function. So heart, lungs, or the respiratory system, and the brain, these are three main organs. It is irreversible and death is defined under 46 IPC. This is now outdated. So absent carotid pulse, no breath sounds, absent of pupillary reflex. Absent carotid pulse, no breath sounds, and absence of pupillary reflex can be considered death, which is a somatic form of death. Okay, somatic death, and also known as clinical death. Okay, clinical death. Okay, so somatic death, that is absent carotid pulse, no breath sound, and absence of pupillary reflex can be considered death or somatic death. So absence of circulation, respiration, and brain function is known as somatic death or clinical death, right? Okay, so we have two types of deaths basically: somatic death, clinical death. Okay, this is one type, and the second type is called as molecular death or cellular death. Okay, so two types of deaths are there. One is called as the somatic or clinical death, and second one is called as the molecular or cellular death. Okay, this time gap between the somatic death or clinical death, where there is a stoppage of circulation, respiration, and brain function, and the molecular death, where the cellular death occurs, that is, the individual cells are dying, is called as the supravital period. This time gap is known as the supravital period. That is the time interval in which person is dead but individual cells are alive. Okay, the person as a whole is dead. Person as a whole is dead, but individual cells, they are alive. Individual cells are alive. In this period many important things occur. That is, first is Jaskow's phenomenon. Jaskow's phenomenon means if you tap on the quadriceps muscle, then there is a knee jerk also. Okay, so imagine a dead person showing a knee jerk reflex. Okay, so this in supravital period, the person is dead but individual cells are alive. So Jaskow's phenomenon is exhibited, is positive in supravital period, where if you tap on the quadriceps muscle, there is a knee jerk reflex. If you apply the drugs on pupil, there will be a pupillary response also. And this period is the best period for cadaveric organ transplantation, where individual cells are alive but the organism is dead. Okay, tripod of life. I am sure I have talked about this tripod of life in our session 1 of marathon. Session 1 of marathon was on your basics of forensic medicine, law, ethics, negligence, and all. This is an important part. Tripod of life is comprising of respiration, circulation, and brain function. This is the tripod of life given by Bichat's tripod of life. Okay, this the tripod: heart, brain, and the lungs. Heart, brain, and the lungs are there. Heart, circulation; brain function; lungs, respiration. Okay, heart, circulation; brain function; and lungs do respiration. And life is comprised of these three things: heart, lung, and brain. Okay, if any one limb is broken, the person dies. So this tripod you have seen three limbs, and here you mount your phone. So this is your life, and these are your three legs: your heart, brain, okay, and the lung. Heart, brain, and the lungs. So if any limb is broken, if heart is stopped, brain and lungs are working, then also person can die. If brain stops, then person can die. If lung stops, then person can die. Okay, this Bishop's tripod of life given by Bishop or Bichat. Okay, so and this in which the person is entering into the death. The person is entering into death is known as atria mortis or gateway of death. Atria mortis or gateway of death. Atria mortis, okay, or gateway of death. Modes of death. What are the modes of death? I told you that whenever there is a loss of function of respiration, circulation, brain function, the mode is basically asphyxia, syncope, and coma. Okay, person can die of asphyxia, syncope, and coma. So these three are the modes of death. Causes of death is basically the disease. Okay, I'm sure I have talked about this in the first session. Causes is basically the disease. Mode is basically how the person died, like asphyxia, syncope, and coma. And manner of death is basically homicidal, suicidal, accidental, all of this. Apparent death. Apparent death in which person appears to be dead but is not dead. The person appears to be dead but he or she is not dead. Also known as suspended animation. Temporary stoppage of life signs. The signs of life temporarily stops, and this is known as suspended animation. The person appears to be dead but it is not dead. Basal metabolic rate, the BMR is so low, is very terminally low, so that clinical signs cannot be detected. Person can again come back to life if there is CPR or resuscitation. Conditions in which suspended animation is seen. In which condition suspended animation is seen? You can get a short note on suspended animation. So be sure to remember it. Apparent death, person appears to be dead, not dead, temporary stoppage of life signs, BMR is very low, so clinical signs cannot be detected. Person can again get back if he or she is resuscitated. Which condition suspended animation is seen? In new HDTV, this is the mnemonic. I is induced or iatrogenic, given by drugs or some intervention. Induced, it is induced animation. N is neonates, E is electrocution. Okay, knee is neonates. Basically, neonates, it is a physiological response. In neonates, it is a physiological response. A suspended animation, this sometimes so the most common cause of suspended animation. In case of electrocution, W is basically wasting diseases like TB, cholera. Okay, wasting diseases, but there is a muscle wasting, or there is muscle wasting, there is decrease in BMR. Okay, in hypothyroidism also. Hypothermia, heat strokes. Okay, hypothermia, heat strokes. Okay, excess, you can remember like this, extreme increase in temperature, decrease in temperature. So heat strokes, hypothermia. D is drowning, T is typhoid, and V is voluntary practice. Some yoga practitioners, okay, they can practice shavasana in that way that they can fake death also. Okay, so voluntary practice or yoga, this can also produce a suspended animation. So this causes are very important. So premature certification of death is very punishable. Please keep in mind the suspended animation part whenever you are certifying death. These are your risk factors, these are your clues, so that you can don't pronounce death prematurely. Okay, this is very important. If you pronounce the death prematurely and the person gets up again from the suspended animation, then you are dead. Okay, so in new HDTP, in is induced or iatrogenic, N is neonates, E is electrocution, W is wasting diseases like TB, cholera, all of this. Okay, H is basically hypothermia and heat strokes. Okay, D is basically drowning, TV is typhoid, and V is voluntary, that is yoga practitioners. Okay, so that's all in death. Now let's move on to the next topic. Okay, now you understood death. Now let's understand how you certify death. Okay, how you certify death. You certify death. Okay, now you understand how we'll estimate the time since death. How will estimate the time since death? How much time has passed whenever the organism has died? Okay, so time interval between death and the time of examination is called estimation of time since death. Okay, time interval between death and time of examination is calculated using thanatochemistry and thanatomicrobiome. Thanatochemistry and thanatomicrobiome, that is the microbiology of the person who is dying, and the microbiology and the chemistry of the person, the physiological, the chemical states of the person who is dying. Okay, so estimated from physical changes noticeable in the dead body. So we'll notice some physical changes in the dead body. Okay, we'll notice some physical changes in the dead body, and from which we'll get to calculate the time of death, the time since death, which is called TST. Okay, so as an autopsy surgeon, this is an important part. So we'll see changes in eye, algor mortis, rigor mortis, putrefaction. Okay, changes in eye, algor mortis, rigor mortis, and putrefaction. Okay, so let's discuss from eye. Okay, we'll go from the eye. Okay, I'm sure you must have heard about 3 to 6 hours. When the 3 to 6 hours, okay, is the early stage, there is a desiccation of eyeball, due to which there is some reddish discoloration of this sclera. The sclera dries up and forms some reddish discoloration like this, which is called as tachenoid deformity. We will read about this tachenoid in the next session. 4 to 8 hours, IOP decreases, okay, intraocular pressure decreases because there is desiccation of the eyeball. The water contents are evaporated. 6 to 8 hours, the cornea gets hazy, okay, you cannot see, the cornea gets hazy and white. And 0 to 2 hours, there is a Kevorkian sign also, which is, I forgot to put over here. Okay, so first one is the Kevorkian sign, that is from 0 to 2 hours in the early stage. Okay, Kevorkian sign is basically whenever you see the retinal blood vessels, the blood columns are fragmented like this. Not blood vessels are not fragmented, the blood vessels are basically becoming torches, and the blood there stops circulating, right? So they get fragmented like this. So fragmented or segmental blood columns, segment of retinal blood within the vessels. Okay, and in vitreous humor, the potassium level increases, okay, due to there is a increased IOP, the decreased IOP also, okay, because there is osmolarity increase, right? Okay, so Kevorkian sign is the first one, Kevorkian sign, that is from 0 to 2 hours. Then you get your tachenoid deformity, that is from 3 to 6 hours. And then you get your haziness of cornea, 6 to 8 hours, okay. And again, it associated increased potassium level in vitreous humor, okay. So this is the thing. Now, post-mortem lividity. We'll understand post-mortem lividity, but first we'll understand the timeline. 1 to 2 hours, there is a mottling of body. 2 to 4 hours, patches. Okay, some patches are there. This is mottling, some patches occur, but these patches, when they join together, they form big patches. Okay, not fixed, but big patches of lividity. 2 to 4 hours. Then 4 to 8 hours, post-mortem lividity gets fixed. Okay, this post-mortem lividity is a pooling of blood, it get fixed. More than 8 hours it persists, okay. Algor mortis, average rate of fall of temperature of the body is 0.4 to 0.7 degrees Celsius. This is the average rate of fall of body temperature of a dead body, okay, average rate of fall of body temperature of a dead body. Now rigor mortis, you understand the rule of 12, right? In 12 hours, algor mortis starts developing. It commences 1 to 2 hours after death. Okay, 1 to 2 hours after death. Death occurs in 1 to 2 hours. Rigor mortis starts. Then it increases, or it appears. Okay, up to 12 hours. It remains stationary up to 12 hours. It remains as it is up to 12 hours of death. And in the next 12 hours, it disappears. Okay, it disappears. So rule of 12 is what is important in case of rigor mortis. Okay, now putrefaction is basically 12 to 24 hours. There is a greenish discoloration of cecum. Okay, the cecal bacteria basically cause a greenish discoloration, and the flank also, but basically of cecum. 24 hours, marbling commences. 36 to 48 hour, marbling is prominent, okay, and 48 to 72 hour, rectum and uterus are basically protruding outer body, okay. 3 4 5 hair nail teeth. Okay, you can go from the softest to the hardest. Softest is the hair, then nail, then tooth. Okay, you can understand by the hardness consistency. 3 4 5. So third day is loosening of hair, fourth day loosening of nail, fifth day loosening of teeth. Okay, third day loosening of hair, fourth day loosening of nail, fifth day loosening of teeth. Okay, so this is the important part which you have to understand. And marbling is 24 hours. 36 to 48, marbling is prominent, and 48 to 72, rectum and uterus protrude. This is not much important. You can remember this 24 hour marbling commences, and 12 to 24 hour greenish discoloration. Adipose year takes 3 weeks to 3 months. Okay, adipose year. we'll discuss. Mummification is basically in dry, dehydrated conditions. The mummies are formed, right? 3 to 6 months. It takes 3 to 6 months. Okay, so adipose here and mummification. This is 3 weeks to 3 months, and this is more than 3 months, up to 6 months. Okay. Okay. So insect activity, 18 to 36 hours. First lay eggs, then 48 to 72 hours the maggots are formed, maggots. Then 4 to 5 days pupa, and 5 to 7 days is basically flies. Okay, so in a dead body you will get maggots, get pupae, get flies. Okay, you can remember this easily, that egg, egg. So 3 letters, 3 days, you can remember, not 3 days, basically 36 hours. Okay, you can remember 36 and egg, 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 3 letters. Okay, you can remember like this, 18 to 36. Then up to 72 hours, 72 hours means your 3 days maggots. Okay, so remember like this, that 36 is egg, 3 days is maggots. Okay, 4 to 5 days pupa, 4 letters, 4 to 5, and 5 to 7 flies, flies, 5 letters, so 5 flies. Okay, remember, easy to remember. Yes. Now, okay, now stomach contents. Stomach contents is from the state of digestion of food and the quantity of food substance. You can estimate the time since death. What is the sequence? Carbs leave earliest, then protein, then fat. So if a dead body is old and has more of fats, okay, it is the old one. If the dead body has more of carbohydrate, it leaves the earliest. Okay, if it leaves the earliest, body is not that old, right? So 1 to 2 hours light food leaves stomach, 3 to 4 hours medium sized meal leaves stomach, 5 to 8 hours heavy meals leave stomach. Usually stomach is emptied in 4 hours, but in case of heavy meal it may take 5 to 8 hours also. Okay, so if the stomach is completely empty, you can very well tell that the body is within 4 hours. Okay, the body is post 4 hours, you can say post 4 hours. Intestinal contents, 6 to 8, 12, and 12 to 18. Okay, look at this. This is our large intestine. This here liver is there, here spleen is there, right? Spleen is there. This is the hepatic. Okay, just a minute. Okay, this is the hepatic flexure, hepatic turn or hepatic flexure. This, let me make you understand by drawing itself. Okay, this is a colon and this is a hepatic flexure, this is a splenic flexure, this is a sigmoid flexure. Okay, if the food is in hepatic flexure 6 to 8 hour, splenic flexure 12 hour, and sigmoid flexure 18 hour. Okay, so 8, 12, 18, you can remember like this: 8 hours, 6 to 8, 12 hours, and 18 hours in this flexures. Okay, this is a intestinal content is there, right? Okay, now contents of urinary bladder. Average rate of excretion is 1 ml per minute. 1 ml per minute is the average rate of excretion of urine. Amount of urine may give indications about time since death. Now bone marrow changes. 1 hour neutrophil nucleus in marrow starts swelling. Okay, the nucleus of neutrophil starts swelling in 1 hour. 4 to 5 nuclei become round, and 10 to 12 hour nuclear rupture. Not much important. Facial hair growth is average growth of hair after shaving. If the person is completely shaved, so how much after the hair is growing? 0.4 mm per day. 0.4 mm per day. You can measure the length of the facial hair and determine the time since death, but this is a very rough idea, and the hair growth doesn't occur after death. Okay, after death there is no hair growth. Basically, if family members are telling that the person saved 2 days ago, okay, and now the person has died, and now you get a dead body with 0.8 mm hair. 0.8 mm facial hair. So you can very well tell that this is consistent with the findings. So this is also not substantial evidence. This is not a substantial evidence. This is also corroborative evidence. Corroborative means helping evidence. Kidney changes within 12 hours. Normal architecture is there, but in 24 hours it is swelling gross swelled kidney, and 34 hours marks and diffuse changes. Blood. So in blood, potassium sodium and chloride level fall, potassium and magnesium level rise. And what other things rise after death? Lactic acid, creatine, NPN, amino acid. Lactic acid, creatine, okay, NPN and amino acids, these all rise after death. Okay, now circumstantial evidence, you can get another things which may indicate your death time since death. Letters, a diary, cinema ticket, degree of coagulation of milk, okay, how much milk has coagulated in his house. Staleness of food, you have seen CID, right? If you have seen CID, they inspect everything, okay, about milk, about newspapers, okay, about newspapers, in which date it was last given, something like that. Dress, wristwatch may stop if drowning occurs, okay, wristwatch may have stopped some days earlier, so that may be determined. Unread newspapers, I told you. Letters, diary, cinema ticket, degree of coagulation, staleness, dress, wristwatch, unread newspapers, these are circumstantial evidences which may indicate the time since death. So in this session we have many timelines. Some important timelines which I want to mark you again is basically, first one is the Kevorkian sign. Kevorkian sign is basically 0 to 2 hours. Then Tachynoir I told you is 3 to 6 hours. IOP decreases in 4 to 8 hours, and this potassium level increases in eye. Haziness occurs at 6 to 8 hours, out of which the Tachynoir is very important, Kevorkian sign is very important. Postmortem lividity, I told you 1 to 2 hours there is mottling, 2 to 4 hours there is coagulation, then 4 to 8 hours there is PML, okay, it get fixed in 8 hours, around 8 hours, and more than 8 hours it persists till putrefaction, okay. And here the mottled appearance is important. Algor mortis, average rate of fall you have to remember 0.4 to 0.7 degree Celsius. Rigor mortis, I told you, rule of 12 you have to remember. 12 hours appear, 12 hours persist, 12 hours disappear, and it starts 1 to 2 hours after death, 1 to 2 hours after death, okay. Then putrefaction, I told you, greenish discoloration 12 to 24 hours. This you have to remember, greenish discoloration of cecum 12 to 24 hours, okay. And marbling starts more than 24 hours, okay, and prominent about 36 to 48 hours. So you can like say that 24 to 48 hours marbling occurs. Then I told you this timeline you have to, very important, 3, 4, 5. Then in the sequence of hardness, hair, nail and tooth. So 3 days hair, 4 days nail, 5 days tooth. What happens? Loosening. Then adipose hair and mummification, this is also very important. Adipose hair 3 weeks to 3 months. Adipose hair is 3 weeks to 3 months, and mummification after that 3 months to 6 months, okay. Then insect activity, what trick I told you, egg basically 3 letters, so 36 hours. Then egg, after egg maggots form at 3 days. Then pupa at 4 days, okay, 4 to 5 days. Then pupa, then flies 5 letters, 5 to 7 days, okay, this is important. Then stomach contents from state of digestion, and this, okay, what thing you have to remember, that stomach empties in 4 hours. Light food 1 to 2 hours, okay, 3 to 4 hours medium sized meal. Then 4 to 5, or you can say 5 to 8 hours heavy sized meal, okay. But here you have to remember that usually stomach is emptied in 4 hours. It may take 5 to 8 hours in case of heavy meal. Up to this is important. Intestinal content, I told you 8, 12 and 18. 8, 12 and 18. Here you add 6. So 6 to 8 at hepatic flexure, 12 at splenic flexure, okay, and 18 at sigmoid flexure. Content of urinary bladder, I told you, the average rate of excretion is 1 ml per minute. Okay. This changes 1 hour. Neutrophil nucleus, nuclear become round, nuclear rupture. This is not much important. Facial hair growth, remember this rate 0.4 mm per day, okay, 0.4 mm per day. This is the thing. Kidney changes, normal architecture, gross marked diffuse changes, not much important, but you remember that 24 hours the kidney swells off. Okay. And blood just you remember this levels and what increases after death, that is lactic acid, NPN, okay, creatine and amino acids. Lactic acid, amino acid, creatine and NPN, nitrogenous wastes. Okay. Then circumstantial evidence, I told you, letters, diary, cinema ticket, all of this you can see. Okay, so that's all in time since death. Let's move on to the next part. Okay, so what happens after death is what known as post-mortem changes. Post-mortem changes. Okay, so post means after, mortem means death. So what happens after death are post-mortem changes, and here we have immediate change, early change and delayed change. Immediate change, early change and delayed change. So what happens immediately? Okay, first immediately after death some changes occur like cessation of circulation, cessation of respiration, loss of muscle reflexes, loss of light reflex. Okay, this is immediate change, circulation, respiration, light reflex. Okay, all of this. Early changes are seen few hours after death till 3 days. So few hours to 3 days are what known as early changes. Now what changes we have? First one is the skin change, eye changes, then all mortis, algor mortis, rigor mortis, post-mortem staining occurs, marbling occurs, and greenish discoloration of right iliac fossa occurs. Right iliac fossa where cecum is present, greenish discoloration. Okay, so we have read the timeline. Now it will be easy for us to correlate the changes accordingly. Okay, so till 3 days whatever changes we have read come under the early changes. So skin changes, eye changes, algor mortis, rigor mortis, postmortem staining, marbling and greenish discoloration of RIF. Skin changes, what happens to the skin after that? Earliest of early changes seen in the dead body. So first organ system you can say to undergo changes in a dead body is always the skin. What happens to the skin? Loss of temperature of the skin, loss of elasticity of the skin, loss of turgor of the skin. Okay, so there is a loss of elasticity, there is a loss of turgor, there is a loss of temperature of the skin. Eye changes, the eyeball sinks up. The eyeball, there is a desiccation of eyeball. There is a desiccation. The correct term is desiccation. So there is a sinking or desiccation of eyeball due to loss of intraocular pressure. IOP decreases. Due to this decreased IOP, the fluid exerted by the eye on its orbital wall, basically it is lost. So there is a sinking in of eyeball. Decreased IOP is 50% in 30 minutes after death, almost zero in 2 to 4 hours. Okay, within 2 to 4 hours your IOP drops to zero. IOP drops to zero. Okay, and it decreases 50% in 30 minutes after that. So you can do tonometry. This tonometry can reveal that there is a decreased IOP value of 50%. Okay, then we'll see another thing which we already saw in that technoidea sclerotica, which I told you, the technoid deformity. Technoid deformity is a reddish brown discoloration across the sclera. So this is a reddish brown discoloration across the sclera in a triangular fashion like this. Okay, it is independent of the etiology of death. This is due to drying of the eyeball or desiccation of the eyeball. Okay, drying or desiccation of the eyeball. Increase in vitreous humor occurs. Okay, this is a important biochemical tool for estimating time since death. This technoid deformity I told you appears after 3 to 4 hours of death. Okay, I'm sure you have watched the previous part of this lecture, that is the timeline I'll be using whenever the event comes. Increase in vitreous humor potassium concentration, I told you, the important biochemical tool for estimating time since death. Okay, then Kevorkian sign or cattle trucking sign. This is an ophthalmological sign where retinal vessels, they are alive, they look like this, Okay, but after some time, after death, basically the retinal vessels get dilated. There is a dilation of retinal vessels having stagnated columns of blood. Okay, the retinal vessels get dilated and the blood columns get stagnated here like this. Okay, so dilated tortuous retinal vessels having stagnated column of blood is what known as Kevorkian sign or cattle trucking sign, and this represents a stoppage of the circulation of inside the retina. Okay, the retinal vessels are basically stopping their circulatory power. Then algor mortis, post-mortem cooling of the body is called as the algor mortis. Post-mortem cooling of the body is known as algor mortis, and here the rectal temperature is basically considered because we have a surface temperature of the body and we have a core body temperature. This core body temperature is what is easily measured in the rectum, that is rectal temperature. Okay, so temperature of the body is lost in three factors: conduction, convection and radiation. Okay, so body loses heat from surface to the extended surroundings, external surroundings in three fashion: conduction, convection, radiation. Core of the body loses its heat via conduction to the surface, which further loss to the environment. Okay, so loss of rectal body temperature occurs. So we have a graph of algor mortis which you have to discuss and is very important. This is sigmoid graph or a inverted S-shaped graph. Sigmoid or inverted S-shaped graph, something like this. Okay, something like this. So we have three phases here. Look at this graph. We have three phases here. Normal body temperature 37 degrees Celsius, all agree. Phase 1 is basically a stationary type of phase. The body temperature is not that is a constant up to a certain period of time. Phase 2, there is a logarithmic decrease in body temperature, and phase 3, there is again a stationary phase. Okay, but the body temperature is maintained at a lower level. Okay, so here in algor mortis graph we are taking temperature versus time. So three phases: phase 1, phase 2 and phase 3. Okay, so temperature, this is the time, and these are your phases. Phase 1 maintained at 37, phase 2 logarithmic decrease, phase 3 is your normal stationary again. So phase 1 is representation of establishment of rectum losing its temperature through conduction to the surface. Okay, so in here it is starting, it is just starting, it is the establishment period where there is the rectum starts losing the temperature through conduction, but there is no change in the core body temperature in this phase. Phase 2, body loses temperature very fast, the very fast stage. Third stage is equilibrium with the external environment. Here it again gets back to the equilibrium at a lower level. It is slightly lower temperature, it maintains its equilibrium and body will not lose any more temperature in the phase 3. So this is phase, the algor mortis graph. Okay, and algor mortis is known as the post-mortem cooling of the body where there is a loss of rectal temperature, okay, via conduction. Okay, what factors affect the algor mortis? What causes the organism or a dead organism to lose temperature more fast or more slow? Right, so age, whenever, remember, there is extremes of age: old age, very young, very old, very young. All of this basically extremes of age cool faster. Built, lean and thin bodies cool faster. Okay, if the thinning of body is a lean and thin body, they cool faster. Clothing, naked bodies will cool faster. Okay, environment, body cools faster in colder environment. In colder environment the body cools faster, the losing of heat is more. Surface on which body is lying, surface also determines or plays an important role. So age, build, clothing, environment and surface, these are the factors which are basically helping, okay, in the cooling process, right, or affecting the algor mortis: age, build, clothing, environment and surface. Okay, so if the body cools slower, the graph is shift towards the right shift. If the body cools slower, if the body cools faster, then there is a left shift. Left shift means the body is cooling faster. Okay, muscle changes. Primary relaxation of muscle is due to loss of muscle tone. Okay, the tone of muscle is lost, but rigor mortis is due to depletion of ATP. Whenever there is a depletion of ATP, the muscle is contracted, but there is no sufficient ATP to cause the relaxation again, because the person is dead. There is no ATP synthesized. The electron transport system is not working now. Okay, so ATP levels and rigidity. When there is a decrease in 15% of ATP, the rigidity or the rigor mortis begins, and there's when there is a decrease in 85% of ATP, there is a peak rigidity period, a peak period of rigidity. Secondary relaxation due to denaturation of muscle proteins. Okay, so primary relaxation is there and secondary relaxation is there. Primary relaxation is due to tone loss. Secondary relaxation is due to denaturation of muscle proteins, and between this is the rigor mortis if in a dead person, and this is due to depletion of ATP. This thing you have to very well understand. Now, Jaskow's phenomenon, when the patella is stopped, I told you that in the supravital period, which is the supravital period in which there is the somatic death and the molecular death period. In somatic death and the molecular death, supravital period, whenever there is a very good period for the blood, you can say organ transplantation, Jaskow's phenomenon is positive, light reflex is positive. In Jaskow's phenomenon, patella is tapped. This tapping leads to the twitching of the quadriceps muscle. Okay, because the primary relaxation phase, in the primary relaxation phase, the contractile unit is intact now also. Okay, but in secondary relaxation phase, there is no Jaskow's phenomenon at all. Okay, so in primary relaxation, basically the Jaskow's phenomenon you will get positive, but in secondary relaxation, whenever there is a complete denaturation of your muscle proteins, there is no Jaskow's phenomenon at all. Okay. Now let's discuss about rigor mortis, which is a very important short note in your FMT exams, and important for this many things, important for your PG also. Development is a gradual process. Development of rigor mortis is a gradual process, right? And rigor is the rigidity term for rigidity, mortis is death. So rigidity after death, okay, appreciated about 2 hours after that. I told you it sets in from 1 to 2 hours after death. Happens after initial primary relaxation phase of muscle. So primary relaxation and secondary relaxation, I told you between that your rigor mortis basically occurs. It follows two rules. One is the Nisten's rule, one is the rule of 12. Two rules are followed by rigor mortis: Nisten's rules and the rule of 12. Nisten's rule and rule of 12. So this is rigidity of death or the rigor mortis. You can see the rigidity, you can see the posture. There is a flexion here, there is a flexion here, there is a flexion here. Okay, so this is a stiff body and this rigor mortis has set in. Nisten's rule. What is Nisten's rule? Rigor mortis goes away from the dead body in the same fashion and it appeared in it. So the rigor mortis appears in a sequence and disappears in the same sequence. So top to bottom fashion, it appears in the top to bottom fashion, it will disappear from the top to bottom fashion. Okay, so heart develops the rigor mortis at the earliest 1 hour after death. I told you rigor mortis takes 1 to 2 hours after death, and this 1 hour is basically for heart. Okay, so heart develop rigor mortis at the earliest. This is the first exception. Earliest external muscle to develop rigor mortis is eyelid. Eyelid is the first muscle, external muscle to develop rigor mortis. Okay, so it starts from top to bottom, right? So topmost is the eyelid. Exception number 2, last to body to get rid of rigor mortis are the digital muscles of the hand. So last one to develop or get rid of rigor mortis. You must be thinking that toes must be the last. Because they are the terminal part, but this is an exception here. The exception is that the digital muscle, the muscles of the fingers, the abductor pollicis, longus, everything you read in your anatomy. Digital muscles of hand are the last muscle of the body getting rid of a rigor mortis. Earliest external muscle to develop rigor mortis is eyelid. Last muscle to get rid of rigor mortis is hand. Earliest organ or earliest part of your body to develop rigor mortis is your heart, that is 1 hour after death. This is your Nisten's rule, exceptions to Nisten's rule, right? Rule of 12, which I already explained to you. 0 to 12 hours rigor mortis develops and peaks in a muscle. Okay, so rigor mortis develop in the first 12 hours, 0 to 12 hours. 0 means 1 to 2 hours have already passed. We are talking about the first part when rigor mortis starts developing. So I am writing 0, but this is again 1 to 2 hours after, because rigor mortis takes 1 to 2 hours after death, right? So 0 to 12 hours rigor mortis develops and peaks in a muscle, which is called a stage of development. Then it persists for 12 hours, so this is a stage of persistence. Okay, the virus muscles in different intensity, stage of persistence. Then 24 to 36 hours it declines like this, which is called state of disappearance. Rigor mortis gradually disappears from the body, which is called the stage of disappearance. And more than 36 hours rigor mortis is completely absent. Okay, so rigor mortis 12 hours appear, 0 to 12, then 12 to 24 it persists, then 24 to 36 it disappears. More than 36, no rigor mortis. Rigor mortis starts 1 to 2 hours after death. 1 hour means heart is the first muscle, which is the last to get rid of digital muscle, which is the first external muscle to have a rigor mortis is the eyelid. Okay, all these things you have to understand. Conditions simulating rigor mortis or look alike conditions of rigor mortis. Some conditions look like rigor mortis, but they are not rigor mortis at all. So some conditions look like RM, but they are not RM. Okay, for example, cadaveric spasm, heat stiffening, cold stiffening, gas stiffening. Okay, so one spasm is there, 3 stiffenings are there. So in stiffenings, heat, cold and gas are there. Okay, easy to remember. Okay, look alike conditions of rigor mortis. Cadaveric spasm. Cadaveric spasm is the complete loss of ATP from a group of muscles before death. Okay, whenever there is a complete loss of ATP, complete loss of ATP before death, okay, this you get a cadaveric spasm, and this in a group of muscle. Rigor mortis occurs in an individual muscle, but this cadaveric spasm occurs in a group of muscle. Okay, it means in contracted state post death. Okay, so they ask you sometimes that what are the differences between the cadaveric spasm and your rigor mortis. Okay, so how cadaveric spasm is different? Cadaveric spasm, remember, is an immediate one. It occurs at the moment of death. It occurs at movement of death. At movement of death. Okay, it is not like rigor mortis. Rigor mortis is basically occurring 1 to 2 hours after that. Okay, so right, in rigor mortis appears after 1 to 2 hours, after 1 to 2 hours of death. Okay, it occurs at the moment of death. Another thing, cadaveric spasm is only affecting those muscles which are active before death. Rigor mortis occurs in all muscles. Rigor mortis occurs in each and every muscle of your body, but this cadaveric spasm occurs in only those muscles which are active just before death. Let's say the person is drowning. Okay, he catches hold in a bid to save himself. He catches hold of some stick. He hold, some leaves he held. Like he's in a state of turmoil, right? So he doesn't know what he is doing or what he or she is doing. So he gets hold of a stick, he gets hold of a leaf, let's say. Okay, and death occurs. So this cadaveric spasm is there. If you check the hand of the person after death in autopsy, you will get the sticks, you will get the leaves, right? So that is cadaveric spasm basically. So it occurs only those muscles which are active before death. He was moving his hand muscle, so cadaveric spasm will occur in your hand muscle only. It occurs in a group of muscle basically. Okay, so this is the thing. So you can say that this is localized one. This is a localized phenomenon. Okay, this is thing. And this is a basically the mechanism is basically neurogenic psychomotor phenomenon. Rigor mortis is a biochemical phenomenon because depletion of ATP is occurring. This is a psychogenic or neuromotor phenomenon. Neurogenic, you can say neurogenic phenomenon. Okay, neurogenic phenomenon, and it involves only voluntary muscles are involved. It doesn't involve any involuntary muscles, but rigor mortis involves all. It involves all muscles. Okay, this is the thing. So this thing you have to remember. Okay, so cadaveric spasm is done. Now we come to heat stiffening. In heat stiffening, basically the temperature is more than 65 degrees Celsius, then there is a coagulation of muscle proteins. If a higher temperature is there, the muscle proteins basically coagulate, which leads to heat stiffening. Cold stiffening, likewise, basically body is kept in a cooling temperature in a mortuary in a very cold temperature. The fluid of the body solidifies and crystallizes. Okay, the solidification of fluid occurs and crystallization also occurs. And this is cold stiffening. Gas stiffening is due to bacterial decomposition of dead body. The bacteria, they decompose the dead body, they inflate the abdomen of the person due to expansion of these gases. Okay, the abdomen and all inflate and also can cause this rigidity type of thing. So gas stiffening is there. Okay, now comes post-mortem staining. Post-mortem staining, also known as post-mortem lividity, also known or PML, PMS, post-mortem staining, post-mortem lividity, PML, post-mortem hypostasis, livor mortis, LIV or livor mortis, suligation, vivises, darkening of death. Many names are there. Okay, so if they ask you, they will tell you suligation, okay, or sugilation, not suligation, sugilation, sorry, slip of tongue, sugilation, okay, or vibices or vibices, whatever you can pronounce, vibices, okay, and ask you a long question based on that. So you don't know this term, then it will be your loss, right? So you have to remember all the terms: postmortem hypostasis, lividity, livor mortis, sugilation, vibices, and darkening of death are all the terms of postmortem staining or livor mortis. So here you have a discoloration, here you have the pale areas. So pale areas occur in your dependent part of body. Okay, discoloration of the dependent part of body. This is the part of discoloration. This is the normal body. This is the normal body. You get paleness where you get paleness in the buttocks and the scapular region. You get paleness and discoloration of dependent parts of body due to peripheral migration of blood towards the skin. Towards the skin, there is a peripheral migration of blood occurring. Okay, towards the skin, the peripheral migration of blood, due to which there is a discoloration. There is a discoloration of dependent parts of body. Okay, discoloration of dependent parts of body. So there is a gravity pooling of blood. There is a gravity pooling of blood occurring. Gravity pooling of blood. This term you have to write. And pale areas where they are seen, buttocks and the scapular region. And phenomenon is also known as contact flattening. Not seen on pressure points. Importance. What is the importance of any livor mortis or post-mortem staining? So position of body after death is a very important thing which they usually see. In still water, post-mortem staining develops on the front of the body. Okay, if there is a still water, okay, in still water only, in drowning in still water, post-mortem staining where it develops in the front of the body. Post-mortem staining cannot generate in the fast flowing water. In case of flowing water, in the case of water is flowing, the post-mortem staining cannot be generated. And in case of hanging, if a person is hanged, what is the dependent part? Gravity, where gravity will act most? At the hands, at the feet, right? So here there is a globe and stocking pattern. So in case of hanging, basically there's a globe and stocking distribution of the post-mortem staining. Then position of the body, then time since death, and cause of death. You can get to know like this: globe and stocking, then it must be a case of hanging. If there is a front of the body, it may be a case of still water drowning. Okay, like that you can position of the body, you can get to know over dependent parts, time since death, and the cause of death, right? Look at this, the timeline which I previously showed you. Let's read it again. 1 to 2 hours there is mottling. 2 to 4 hours there is not fixing but patches are coalesced. 4 to 8 hours there is development and fixation, and more than 8 hours it persists till putrefaction sets in. So you can remember 1 to 2, 2 to 4, 4 to 8, more than 8. Okay, so 1 to 2 only patches occur or mottling occur. 2 to 4 there coalition of the patches. 4 to 8 hours it develops and fixed, and more than 8 hours it is completely persisting in the body, right? Okay. So what I mean by fixing or shifting of the postmortem staining. Discoloration, if it fades on applying pressure, then it is called as shifting. It is called as shifting. Let's say here we have postmortem staining and you touch it with your fingers and you press it, you apply some pressure here and it fades, it becomes pale again, then we tell that it is not fixed, it is shifting. It is shifting to one portion to another. Okay, this is shifting. But once you get about 8 hours, around 8 hours, it get fixed. Okay, fixing begins at about 6 hours and complete fixation at 12 hours. So basically about 8 hours I am telling that it is persisting and it is getting fixed. Okay, so fixation begins at 6 hours after death and complete fixation occurs at 12 hours after death. Okay, so this is shifting. This is if the applying pressure and this is fading. Okay, this is shifting. If it get fixed, if you even if you apply the pressure, it will not get faded like this. This is called fixing. Okay, so we have 3 cases to discuss here, and these 3 cases you will get a clarity of your time since death. Look at this. Case number 1, 0 to 6 hours. Case number 2, 6 to 12 hours. Case number 3, more than 12 hours. Okay, 0 to 6 hours, there is no fixation at all. Complete shifting occurs on pressure test. Complete shifting will occur. 6 to 12 hours, there is a partial fixation. Partial fixation means some parts will be fixed, some parts will be meaning like this. More than 12 hours, there is a complete fixation. There is no shifting on pressure test. Okay, so accordingly, if there is a complete fixation, you can very well tell that the dead body is more than 12 hours of age. If there is a partial fixing, then 6 to 12 hours, and no fixing, 0 to 6 hours. This is one timeline, and another timeline which I told you, 2 to 4 hours. Okay, 4 to 8 hours, more than 8 hours. Okay, 1 to 2 hours. 1 to 2 hours you get mottling and patches. 2 to 4 hours the patches are coalescing. Okay, 4 to 8 hours there is like fixing which is starting off. Okay, fixing starting off, right? 0 to 6 hours, and more than 8 hours it is persisting, and after 12 hours there is a complete fixation. What are the different colors of postmortem staining? There's the different colors which may give you hint about what poisoning has occurred in the body. Okay, normal is reddish brown. Okay, normal color of postmortem staining is basically reddish brown. Look at this, reddish brown types of color. Okay, if the postmortem staining is a cherry red color, then carbon monoxide. If it is a brick red color, then cyanide. Black color, then opioid. Brown color, perchlorate. Blue color, aniline dyes. Give an idea of the cause of death also. Okay, so you have to remember some important things. Like normal color you have to remember very well, reddish brown, and 2 other shades of red are there. If it is bright red or cherry red, carbon monoxide, CO. If it is a type of deep type of red or brick red or crimson type of color, then cyanide. Okay, so carbon monoxide and cyanide you remembered. Opioid is black. Okay, perchlorate is brown, and aniline dyes, basically aniline dyes affect basically bladder, right? So color of urine is basically, you can remember something like that, some analogy I don't know. Okay, you can remember this blue. Chocolate brown is not much important. So graphical representation, I told you, this is a graphical representation. This is a rigor mortis. Look at this. First one is a rigor mortis. Pay attention to this graphs, because you can draw this graphs in the theory exams also, and this can be asked in your you can say PG entrance also, because this graphical based questions, what they love. Okay, 0 hours, this is the death, this is the time in which death has occurred, this point. Okay, 1 to 2 hours after, 1 to 2 hours after, 1 hour they have shown, 1 to 2 hours the rigor mortis is starting, and it rising up to 12 hours. Up to 12 hours rigor mortis is being developing, then from 12 to 24 hours it is being persisting. So stage of appearance is there, stage of persistence is there, then up to 36 hours it is now depleting. Okay, so there is a stage of disappearance is there, up to 36 hours. Okay, understood rigor mortis graph. Then algor mortis, I told you, temperature plateaus here from 37 degrees Celsius, then depletes in the phase 2, and then again gets in equilibrium with the surrounding. Okay, then Putrefaction, it starts from 18 hours plus. The putrefaction starts from 18 hours and it rises up to S12. Okay, decomposition, it goes on. It's never ending process because the bacterial action, right? Okay. Now liver mortis is basically what is called as the post-mortem staining or sugillation or viviscus or post-mortem lividity or liver mortis, anything which you can tell. So it's starting from the 6 hours, 0 to 6 hours, there is increase liver mortis, and here there is no fixation here, from 6 to 12 basically there is partial fixation here, there is partial fixation. After that it is basically persisting till decomposition. Okay, it never touches bottom, it persists, but it is persisting till putrefaction basically or decomposition. Okay, algor mortis is the only graph touching the y-axis. Okay, which graph touches the y-axis? Algor mortis, right? Because it is starting at a particular temperature. Okay, now the last early change after that. Okay, another thing, marbling is there. Marbling we haven't discussed, right? Okay, yeah, greenish discoloration of right iliac fossa, which I told you, due to decomposition of organic matter in the gut. The right iliac fossa is the right iliac fossa region. There's a greenish discoloration which you can very easily see in this diagram. Okay, some about in the umbilical part also, but basically over the right iliac fossa, due to decomposition of organic matter in the gut. And this organic matter, this load of bacteria, basically there is a bacteria, this bacteria act upon this you can say this blood circulation is basically altered by this bacteria. Methemoglobin is increased and H2S gas is basically produced. Okay, organic matter, when it decomposed, H2S gas is produced, which is giving a greenish discoloration, because maximum bacteria are present in the cecum part also. Okay, so discoloration is earliest seen in the right iliac fossa after 18 hours after death. Okay, after 18 hours, more than 18 hours it is seen. Okay, greenish discoloration of RIF. Okay, marbling is basically greenish blue discoloration of the vascular epithelium due to reduction of hemoglobin into sulf-methemoglobin. How it occurs? Look at this. Hemoglobin is acted upon, is binding to H2S, H2S is produced by the bacteria, I told you, and from sulf-methemoglobin, which is a violet color. This violet color sulf-methemoglobin is basically staining this blood vessel. If the blood vessels are stained, it will look like this. Okay, so greenish blue or purplish discoloration, you can write greenish blue to purplish discoloration, where the veins, the blood vessels become very prominently discolored. Or look at this discoloration, you can see this discoloration. Okay, this is a greenish blue discoloration of the vascular epithelium due to reduction of hemoglobin into sulf-methemoglobin. This type of greenish blue discoloration occurs. Okay, seen between 36 to 72 hours. Okay, 36 to 72 hours. Okay, look at this timeline again. So I'm coming back to the timeline again and again, because this is important to correlate it to timeline, and I'm sure that you must be facing problem in remembering this. So if you come it back again and again, then it's very easy to remember. 12 to 24 hour greenish discoloration occurs. Okay, so I told you 18 hours, so 18 hours you can take a exact time, you can say. Okay, more than 18 hours it gets greenish discolorated, right? So cecum and right iliac fossa. Okay, and 24 hours marbling commences. 36 to 48 hours marbling is prominent, and 72 hours marbling is gone. So it is prominent between 36 to 72 hours. 36 to 72 hours marbling is prominent. Okay, 36 to 72 hours means this 36 to 72, 3 days, right? Up to 3 days the marbling is prominent. Okay, then 3, 4, 5 loosening of hair, nail and teeth, which we have already read, right? So let's get, so seen between 36 to 72 hours, which I already told you now, right? So it starts from 24 hours, some books write 36 hours, but you can get prominent marbling within 36 to 72 hours. First place of marbling is basically junction of head of neck and the chest. Okay, it's a junction of the neck and the chest, where the shoulder region basically, where the marbling is first seen. Then it moves to the extremities, right? Okay, a phenomenon is there postmortem luminescence. Postmortem luminescence means there is a luminescent glow in the body due to bacteria and fungi. Some bacteria like photobacterium and some fungi like armillaria. Okay, armillaria. So photobacterium and armillaria produce a postmortem luminescence. So this completes our early and intermediate changes, right? So we discussed about skin changes, about the skin turgor, elasticity loss, about all the temperature of the skin. Okay, we discussed about algor mortis and all the graphs related to this. Okay, then eye changes, I told you about tachinoidea sclerotica, right? About the Kevorkins and the cattle trucking sign, about IOP changes. IOP decrease up to 50% in 30 minutes, 50% in 30 minutes, and about 100% in 2 to 4 hours. IOP is decreased, right? We talked about algor mortis, change in the temperature part of the body. So algor mortis graph we saw, it is a plateau phase, then there is exponential loss, then there is a again a you can say equilibrium established. Rigor mortis we discussed in the same fashion. Rigor mortis, I told you, two rules are followed, right? It starts from 1 to 2 hours, it persists till it starts 12 hours, right? In 12 hours it appears, in 12 hours it persists, in 12 hours it disappears. This, okay, rule of 12 is there, the Nistance rule is there. In the, it appears in the same fashion as it disappears, or it disappears in the same fashion as it appears. But we had some exceptions, like the last muscle to the last part to develop rigor mortis is not your toes, is your digital muscles of hand, and the first to develop your rigor mortis is not your eyelid, it is first your heart. Okay, but if we ask about the first external muscle to develop rigor mortis, it is your eye muscle, that is the eyelid muscle, right? LPS. Then postmortem staining, postmortem lividity, postmortem livor mortis or sugilation or viviscus, anything you can call. Okay, this we talked about. And what are the colors? So colors, you saw that carbon monoxide you get cherry red color, cyanide you get brick red color, okay, crimson type of color. In opioids you get black color, okay. In perchlorate you get brown color, perchlorate brown color. Aniline dyes you get blue color, okay. So they may suggest you a poisoning also. It may be important in your whenever you read toxicology, right? And marbling, I told you, marbling is basically prominent from 36 to 72 hours after death, okay, and starts at the earliest point is the junction between the neck and the arms, or neck and the shoulder, which is shoulder portion, okay, and the neck and the shoulders, or neck and the hands where they meet. Okay, so marbling is there due to this sulf-meth hemoglobin, which is staining the vessel wall. And we discussed greenish discoloration of right iliac fossa due to cecum, due to bacteria in the cecum. This greenish discoloration is basically occurring, okay, due to this bacterial action, okay. And lastly we saw some photoluminescence also, and due to any photobacteria or due to fungus like armillaria, okay. So immediate changes, I told you all of this, and early changes we discussed in detail. Now let's move on to the next part. Okay, now we have come to the slightly easier topic of delayed postmortem changes. Basically, whenever there is a decomposition of body, the complex tissues get broken down into simpler organic compounds. So it follows the disappearance of rigor mortis. After rigor mortis, what happens, then the decomposition sets in, or the putrefaction. Decomposition is a general term, may be applicable to wastes, vegetable wastes, animal wastes, but putrefaction is the specific term. And specific term for bacterial effect plus cellular autolysis. So what is the definition? Decomposition of body organs due to bacterial effect or cellular autolysis is known as putrefaction. So putrefaction is the decomposition of the dead body or the dead body organs due to bacterial effect or due to cellular autolysis. So breakdown of cell membrane with release of cytoplasmic enzymes is known as cellular autolysis. And bacteria, because they start decomposing the dead body. It's a very scary diagram, don't get scared, right? Liquefaction of body organs occur basically in putrefaction. The bacteria starts liquefy the organs of the body by cellular autolysis, because they break down the cells, and the cells release some hydrolytic enzymes. These hydrolytic enzymes liquefy the body organs, and there is a foul smell, there is no preservation of body at all. There is no preservation of any architecture of body at all. You can see there is no there is preservation of architecture here, but if it is more decomposed, then this parts will be eaten up by the bacteria. The bacteria will eat up the whole thing, right? Normal time taken for decomposition is 7 to 10 days. This is an important timeline. Okay, and the important thing is that the decomposition basically follows rigor mortis. That is more than 36 hours. This decomposition sets in. Decomposition depends upon the temperature and the moisture. Okay, the putrefaction or the decomposition depends upon the temperature and moisture. If there is a higher temperature, example in summers, so temperature and moisture are the two conditions in which putrefaction depends. If there is an increased temperature in summers, okay, then and moisture no change, increased temperature, more putrefaction, 3 to 4 days. Okay, in summers, so 3 to 4 days for the body to putrefy. In decreased temperature, that in winters, it is slow, 14 to 18 days. Okay, it can take 14 to 18 days. Normally it is 7 to 10 days. Normal time is 7 to 10 days, and in winters it is basically taking double time. In summers, you can remember, it is taking half of time, almost half of time. If there is no change in temperature but there is increased humidity or moisture, then saponification occurs. Okay, saponification is a variant of your decomposition. So if there is no change and increase moisture, then saponification or adipocere formation occurs. If there is no change in temperature but there is decreased moisture means dry and hot fast blowing air, then there is a mummification of the body. So increased hot and moist and stagnant air, saponification, adipocere, and decreased moisture, less dry and hot fast blowing air, mummification of the body, right? Organic rich enzymes autolyze fast. Maceration is the aseptic autolysis of the dead fetus. Okay, whenever there is a dead fetus, there is a aseptic autolysis due to you can say release of enzymes from the cells. There is a aseptic autolysis. Here bacteria is absent. The main thing is that bacterial action is absent, but this is liquor amni. Liquor amni is basically the amniotic fluid. So in the amniotic fluid, there is a aseptic autolysis of the dead fetus. In this maceration, we'll talk about this in infanticide chapter also. In maceration, there is a whitish grayish discoloration of the body, okay, with a disagreeable sweet smell of the fetus. Now, what external changes occur in this decomposition? First is the color change. First change of decomposition is the color change, that is of. So starting off with external changes in decomposition, color changes. First one is color changes. First sign of discoloration is seen in right iliac fossa. I told you that greenish discoloration is due to bacteria in cecum. Cecum has the maximum bacterial concentration, due to which they react with hemoglobin, and this H2S gas produced by this bacteria makes up sulf-methemoglobin. Sulf-methemoglobin basically goes and stains the blood vessel, and marbling is also due to this. Okay, internally discoloration of aortic intima, okay, followed by undersurface of liver. Aortic intima is also, you can say, discolored, right, due to the sulf-methemoglobin. First one is the right iliac fossa, then aortic intima, then undersurface of liver. Okay, so offensive smell is due to this H2S gas only. This H2S gas has a smell which they commonly ask you, and this smell is called as the rotten egg smell. So in decomposition also, skin changes, you will get color changes, right iliac fossa greenish discoloration of aortic intima, and with the color changes, with the smell change, that is offensive smell, rotten egg smell. In India, the changes are seen about 12 hours in summer and 36 to 48 hours in winter. Okay, about 12 hours and 36 to 48 hours in winter. Okay, other changes I told you, hair, nails and tooth get loosened, 3, 4, 5 I told you. Discoloration of face, frothing from mouth, eyeballs protrude out, bulge out, lips get swollen, tongue gets swollen, fish mouth like appearance. Okay, this mouth gets like this, some O type of appearance. Okay, look at this, you will understand this mouth appearance. This O type mouth appearance. You can see the protrusion of the tongue and the swelling of the tongue, swelling of the lips, okay, and protrusion of the eyeballs, swelling of the nose, disfigurement of the face, right, and a fish mouth appearance. This is called as the fish mouthing or fish mouth appearance, right? Okay, so this is there. Discoloration phase, frog from mouth, eyeballs protruded, lips swollen, tongue swollen, okay, all of this. Post-mortem blisters and post-mortem peeling. There is a peeling of the skin, okay, due to this decomposition. Marbling occurs. Distension of abdomen due to gas stiffening, swelling and blue vein stocking appearances, swollen genitalia. Rectal and uterine prolapse also occur. I told you in the time since death thing. Heavy maggot infestation. Maggots, I told you. Let me again revise you the timeline. That is, first you'll get eggs you get at 36 hours, right? Then you get maggots around 3 days, okay, around 3 to 4 days. Then you get flies, then you get pupa you get around 4 to 5 days, okay, and then you get flies 5 days onwards, 5 day onwards. Okay, so this is a timeline of the insect infestation. Maggot holes sometimes may mimic bullet wounds. So one differential diagnosis of bullet wound is maggot holes. Maggot mainly bury inside the skin and produce some holes, and these holes may mimic bullet wounds. Now, internal changes. What are the internal changes in decomposition? These are the external changes which you can very well see. Now let's move on to the internal changes in decomposition. Putrefaction. So parenchymatous organs are more easily putrefied than muscular organs. So muscular organs basically resist putrefaction, but parenchymatous organ putrefy easily. But stomach and small intestine are you can say exceptions, because these are muscular organs. Stomach has muscles, small intestine has muscles, they have stiff walls, still they putrefy easily. Okay, liver gets honeycomb like appearance. This liver get a honeycomb like appearance, that get porosity. Okay, there is an increased porosity of liver, increased porosity of liver. Swiss cheese appearance, foamy liver or honeycomb appearance are the names of soft flabby livers, and blisters are also present. Some areas are blistered, some areas are eaten away like porous. Okay, so honeycomb appearance, Swiss cheese appearance or foamy liver are basically produced. Brain gets soft and decoloration of brain occurs. Heart is white granularity known as miliary plaques. Okay, so if this is the heart, we get some white granularities present, which are plague like deposits, plague like, and these are called as the miliary plaques. Okay, so miliary plaques are produced in the heart. Brain is soft and decolored, and this liver gets a honeycomb or Swiss cheese appearance. Prostate and uterus are the last organs to decompose. In males it is prostate, in females it is uterus, the last organ to decompose, and this may aid in the identification of the gender. If clothing is present, there is a delay in putrefaction, you can understand. Age. Children have a fast putrefaction than old people. Female bodies decompose faster. Fat-rich bodies decompose faster. Okay, whenever there is a fat, there is a good amount of decomposition. Okay, in advanced stage of decomposition, cause of death cannot be obtained except for poisoning, fracture, and firearm cases. Why? Because in advanced stages of decomposition, basically, the skin is totally eaten away. If the skin is totally eaten away, you can get only the characteristic smell or the characteristic, if you send it for toxicological analysis, you can get to know about poisoning. So if totally eaten away, then bones must be present, and this bones fracture can be ascertained. And some firearm cases also, because in some firearm cases, there may be a bone penetration of this firearm, and so firearm cases can also be opined in case of advanced decomposition. Casper's dictum. What is Casper's dictum? Casper's dictum tells that the rate of putrefaction is different in different media. Okay, so a person is decomposing in land, a person is decomposing in water, and a person is decomposing in the earth, in the buried in the earth. If you buried a person, okay, if someone killed and buried a person, then he will decompose the slowest, 8 weeks. Okay, decompose about 8 weeks. And if a person is in water, the decomposition will occur around 2 weeks, and fastest decomposition occurs in 1 week. Okay, in 1 week. So 1 week, 2 week and 8 week, 1 to 8 is the ratio, is according to the Casper's dictum. This may vary. This ratio may not be true in many cases, but this is a dictum that fastest decomposition will occur in the land, the air, in the air contact. Slowest decomposition will occur in the earth or buried in the earth, and moderate time will be taken for decomposition in case of water, right? Okay. Now let's come to adipocere formation or saponification. I told you here, look at this, that saponification will occur whenever there is a increased moisture or increased humidity. Okay, so saponification or adipocere formation, the most important characteristic is basically increase in moisture content. If there is an increase in moisture content, then saponification will be faster, will be basically present. There is no such problem in temperature. Temperature may be high, temperature may be low, it may be summer, it may be winter, but the most characteristic thing which you require for saponification is moisture, right? Increased moisture. So saponification or adipocere formation is a modified decomposition in abundance of moisture. I told you that the characteristic one is the moisture must be present. So always remember, whenever there is a moisture present in decomposition, it is adipocere formation or saponification. And this appearance of saponification is called as rancid butter appearance. Rancid means rotten, or you can say expired. So when butter gets expired, it gets greasy. Okay, you have felt the grease in your bicycles, right? So greasy appearance of body, you can very well get a feeling or get a you can say intuition of this greasy appearance. Okay, I need not explain. So rancid butter appearance or greasy appearance occurs. A sweet smell is there, a ammonical sweet smell is there, sweet disagreeable smell or ammonical smell is there. Soapy and greasy appearance. There is inflammation in some parts. You can see there is some inflammatory lesions which are again saponified. Waxy whitish grayish appearance. Okay, it is a waxy appearance, a greasy appearance, waxy, greasy, white grayish appearance, and soapy appearance with sweet smell. Okay, the smell is basically sweet smell or ammonical smell. Cold adipocere. If there is a cold adipocere, it is hard and brittle. Okay, this greasy appearance occur in basically warmer areas if there is increased moisture as well as increased temperature. But here, where there is increased moisture but temperature is less, then this becomes hard and brittle sometimes. Easily breakable, right? So adipocere takes 3 weeks to generate. So adipocere, the timeline is basically, I told you, 3 days to 3 weeks. Okay, this is for generation, but after generation it takes 3 weeks to 3 months. It remains in that state. Okay, so adipocere takes 3 weeks to generate and completes in about 3 months. I told you in the time since death module, or in the previous section, we saw that adipocere or saponification or adipocere is basically 3 weeks to 3 months. Okay, so it starts about 3 days. It takes for generation about 3 weeks, but it remains persistent up to 3 months. Okay, and mummification, which occurs in less moisture, in dry humid climate. Okay, not humid, in dry desiccated climate. Okay, mummification is 3 months to 6 months. Okay, what is the cause of saponification? Cause of saponification is that hydrolysis and hydrogenation of body fats. So you have some residual fat content in your body. We have some residual fat content, and this residual fat basically undergoes hydrolysis. This residual fat undergoes hydrolysis and hydrogenation, hydrolysis and hydrogenation of body fats, due to which unsaturated fat is converted to palmitic acid type of fats and forms at any site in which fat is more. So obese people have increased adipocere formation. You can see there is more adipocere formation in the buttock area. Okay, in the stomach area. Okay, this upper belly portion. Okay, because there is more fat present over here, right? So more fat, more adipocere formation, and it forms at any site in this fat is more. Obese people has increased adipocere, but features are well preserved. This is an important line, that bodily features, the features of the body. Okay, you can see the hand, you can see the shoulder area, you can see the back. Okay, all these are nicely preserved. Okay, so you can get to know the cause of death very easily from this adipocere body even. Okay, So features are well preserved. Now comes mummification, where features are not preserved but facial features are preserved. Rest all basically get decomposed like this. Okay, so mummification is a modified decomposition in the absence of moisture. Let's go to the previous part. Look at this. Yes, mummification, there is no problem with temperature. Temperature may be increased, temperature may be decreased, right? But here it is moistureless environment. It is a dry, hot, fast blowing air. Dry, hot, fast blowing air, especially in deserts. Okay, in Egypt you must have seen mummies, right? You must haven't seen, right? But you know that when mummies are being talked of, we are talking basically of Egypt, right? So mummification in that type of climate, where there is a dry hot climate, dry hot humid less climate. Okay, there is no humidity, humidity is absent, and the air is fast blowing air. Then this type of body is there, dehydrated, desiccated body, because the 70% of water of total body water is lost. 70% of total body water is lost. Dehydrated, desiccated body. There is no smell, and facial features are preserved. You can see the facial features are somewhat preserved, not completely. This statement is not true completely, but somewhat they are preserved, right? And time for mummification is 3 months to 12 months, some, right, 6 months. Okay, you can take it variable time, but it starts about 3 months to variably, right? Body becomes thin, stiff and brittle. Okay, the body becomes dehydrated, thin. Okay, and the body can't move, so it is stiff. Of course it can't move, but the stiffness is there, a stiffness is there. Then time for mummification 3 months to 12 months. I told you, body becomes thin, stiff and brittle due to extreme temperature. Growth of microorganisms are also not there, because if there are viable tissues present, then microorganisms can get attracted and can grow and decompose them. But here there is no viable tissue at all, right? There is no organic matter present. The body is completely dehydrated, desiccated, right? And skin becomes translucent due to absorption liquefied fat. This fat is also reabsorbed. Okay, fat is you can say liquefied, right? So absorbed and liquefied this fat, and this body becomes very thin. The skin becomes very thin. You can very well appreciate the bony landmarks. You can very well appreciate the pelvic structure, and very well appreciate all these joints and all, right? And chronic arsenic poisoning favors mummification. This is an important line, right? So skin is just of one prominence, I told you. Chronic arsenic poisoning and antimony also, A and A, right? Antimony poisoning can cause severe diarrhea. If there is chronic antimony poisoning, severe diarrhea, there is desiccation and water content decreases, so it favors mummification. And recognition of injury can be made in this case, right? Okay, then comes embalming. Okay, let's read about embalming, then we'll end. Embalming is artificial technique of preserving the body by injecting antiseptic and preservatives. Okay, so we are injecting antiseptics and preservatives, and we are preserving the body. Ideal time is less than 6 hours after death is very effective. If a person is dying at 6 a.m, okay, you preserve the body within 12 a.m, that's a very ideal time. What are the embalming chemicals that are there? Preservatives, we are using formaldehyde, glutaraldehyde. Okay, germicide, we are using phenol. Wetting agent, glycerol. Buffer, sodium carbonate, sodium citrate. And dye is eosin, and diluent is water, 10 liters of water. And ethanol is not a component of embalming fluid. So embalming fluid has some 5 components. One is your preservative, germicide, wetting agent, okay, buffer and dye. Preservative, germicide, wetting agent, buffer and dye. Okay, so these are the wetting agents. Preservative, formaldehyde, glutaraldehyde. Germicide is phenol. Wetting agent, glycerol. Buffer is sodium carbonate, sodium citrate. And dye is basically eosin. Okay, you can remember this like this. PG diploma in West Bengal in WB. Okay, so P is for preservatives, B is for preservatives, G is for germicide, D is for dye. Okay, preservative, germicide and dye, and diploma, and then WB is wetting agent and buffer. So you can use this mnemonic. Diluent fluid is water. Okay, PG diploma in West Bengal. Then types are there. Artificial embalming is the best one. Cavity embalming, surface embalming, and hypodermic embalming. Arterial, cavity, surface, and hypodermic. How to insert? inject electric motor can be used for injection of this fluid. This embalming fluid can be connected to an electric motor. This is a motor, and this may be used to administer the fluid. Best technique is discontinuous injection and discontinuous drainage. Okay, so we are not doing it continuously, we are discontinuously injecting and draining. So we are injecting, then after some time we are draining, again injecting, again draining. You don't need to know much about this, but you just hear. Fluid should be injected high pressure and low volume for better perfusion. Okay, so high pressure to low pressure, high pressure to low pressure, right? And volume must be low according to Boyle's law. So high pressure and low volume are favored. Best vessel is femoral vein. Okay, so femoral vein is more preferred. Then embalming, if it is done in jaundiced person, they have a greenish discoloration. So formalin, it reacts with bilirubin and forms biliverdin. Okay, this breaks bilirubin. Formalin breaks bilirubin to biliverdin. This yellow converts to green, and greenish discoloration may occur. If normal person is embalmed, okay, normal person, then it becomes brown color. Okay, embalming should never be done before autopsy in poisoning cases, because formalin can destroy the poisons, right? And evidences may disappear under evidence may disappear if the doctor is doing embalming before determination of toxic, before the toxicological analysis is done. If he suspects poisoning, again does embalming, then he may be sued according to law. Okay, as per the law, disappearance of evidence, this is now outdated, but you just know that he can be punished, right? So because formalin will break down the poisons, many poisons are broken down by this formalin. Okay, so that was about embalming, and that were about your delayed post-mortem changes, right? Okay, so with that we come to the end of this chapter, that is thanatology. One thing which is not under thanatology, but some miscellaneous things like your autopsy, about the types of autopsy, about the technique of autopsy, all of that, and about infanticide will be dealt to in another session. This session was about the postmortem changes and about the study of death, about the time since death, right? So that's all in today's session. Thank you, and let's meet in the next lecture. Bye.